Okay, so what is a nephron? You can see here in the picture. So this is like tubular structure, the major functional unit of kidney. That's important. Major functional unit of kidney. There's about 1 million of nephrons in each kidney, so you can imagine they're pretty small. So 1 million per kidney. So how do nephrons work? So at the beginning of the nephron, here at a small ball, we're going to draw it in more detail in a second. You have something called the glomerular filtration membrane, and through that membrane, blood gets filtered. So nephron filters blood, and then everything that gets filtered through into the nephron becomes the ultrafiltrate, also called the primary urine. So ultrafiltrate, and there is about 180 liters of ultrafiltrate every day. And obviously, if you were to pee all of this out, then there would be much of you left by the end of the day, by the noon actually. So it is 180 liters, then go through the rest of the nephron, and vast majority, like 99% of the stuff that's in the primary ultrafiltrate gets reabsorbed. So that the ultrafiltrate gets reabsorbed. In ducts or tubules of nephron. Right? Maybe I should say that 99% of the stuff gets reabsorbed in tubules and nephron, and that will give you the final urine. And you might have an idea how much final urine a human being produces per day. It's from 1 to 2 liters. I think it's nice to remember 1.8, because then obviously you know that about 99% of the ultrafiltrate gets reabsorbed in the tubules of nephron, and that 1% will stay. Just to mention, not all of the substances get reabsorbed in the same at the same rate. Some stuff, all of it gets reabsorbed, like for example amino acids, glucose. Some stuff like urea, ammonia, and some ions. Almost all of the stuff that is present in ultrafiltrate eventually will make its way through into the final urine. Right? So the relative absorption of different salts is different. Okay, so now we're going to draw the nephron. Before we start drawing the nephron itself, we have to draw the blood vessels that lead into the nephron and which provide the blood that will be filtered eventually. Actually, you need to know the name of these vessels. However, the name is nothing hard to remember. One blood vessel brings the blood to the nephron. That will be called the afferent arteriole. This is the one I'm drawing right now. And then the one that carries it away will be the efferent arteriole. So this one. Okay. So to label it, you have afferent arteriole. Arteriole is a small artery, and then you have efferent. Arteriole. Okay, and then in the middle of them you have this convolute. Actually, the afferent arteriole branches into smaller and smaller branches. However, that would not bode well on my small drawing. So all of these capillaries that branch from the afferent artery, they make the convolute, this ball of a structure, capillary structure. This is called the glomerulus. So glomerulus. And from the capillaries of glomerulus, the blood will get filtered into the nephron. So now I'm going to draw the first part of the nephron, and that will be the Bowman's capsule. Okay. So this is Bowman's capsule. Bowman's capsule. Okay, and what happens here? So you have the blood coming through the afferent arteriole that starts to branch into small capillaries that eventually will form the glomerulus. The capillaries of glomerulus are quite porous, and so vast majority of the blood that, or not vast majority, but the big chunk of the blood that enters glomerulus will get filtered. Okay, but you know that filters can have different size holes. They don't have to filter everything at the same rate. The same thing with glomerulus. So some things get filtered, some things do not. Most importantly, things that do not get filtered through the glomerulus into the Bowman's capsule are blood cells and plasma proteins. So for example, albumins, immunoglobulins, they do not pass through into the Bowman's capsule. Only substances like water, ions, amino acids, glucose, bicarbonate, they do. They are small enough so they can pass through that filtration barrier. You can imagine it as some sort of like net or web that prevents passage of big stuff and allows passage of small stuff. Okay, so and that's the Bowman's capsule, right? And then all this stuff that gets filtered, like water, ions, glucose, amino acids, that will be present here in the Bowman's space. Okay. You'll learn definitely more detail in histology. For example, you might see that the Bowman's capsule has two layers. One layer adheres to the glomerulus. This would be made up of cells called porocyte. Then you have another layer that sort of like surrounds the all of Bowman's capsule. That would be the Bowman cells, right? Here I draw just as a line, but you definitely can guess that there would be cells located, right, lining it, some epithelial cells. And the same would be for this layer, except here it's way more intricate. Okay, and so this is the first part of the nephron, the Bowman's capsule. Okay. Now the next part of the nephron, this will be the heavy walker of the nephron. This is where a vast majority of the stuff that passes into the Bowman's capsule will be reabsorbed back into the blood. So this is the proximal convoluted tubule. And this is the most metabolically active part of the kidney, proximal convoluted tubule. Okay, and so what happens here? Here you filtered all of that water and ions, etc. Here, vast majority of that will get reabsorbed again. So water, ions, glucose, amino acids. All of this gets reabsorbed in the proximal tubule. Maybe I could write the name right. So proximal convoluted tubule. Convoluted tubule. Sometimes you can also see only proximal tubule. Actually, the proximal tubule could also have like a straight part, but usually you just see it as a proximal convoluted tubule. Okay, so this is where the vast majority of the stuff gets reabsorbed again. And then the, let's say, following parts of the nephron are a little bit more picky, and they play a role mostly in the relative amounts of stuff that will be present in the final urine. So next one would be the loop of Henle. This is arguably the longest part of the nephron, so loop of Henle. Loop of Henle also can have thick and thin part. but I don't think that's that important for your entrance exam. So loop of Henle. Loop of Henle. Okay, and so what it does, again, it just reabsorbs some ions and water, but it does not absorb any of the glucose and amino acids. All of the glucose and all of the amino acids get reabsorbed in the proximal convoluted tubule, and none of these substances should reach the loop of Henle. A notable exception are people with diabetes, because they have so much glucose in their blood that they have very high concentration or high amounts of glucose inside the nephron, such a high amount that the transport proteins cannot keep up, and they just cannot absorb all of that. So one of the possible diagnostic tests for diabetes is that they pee into they pee into some test tube, and then you into that test tube with urine put a dipstick, and that can detect the presence of glucose. And if glucose is present in the urine, that means that most likely they have very high concentrations of glucose in their blood and they have diabetes. Anyway, but loop of Henle. In loop of Henle, water and ions get absorbed. This time it's a bit more selective. You don't need to necessarily know that now, but only some ions are reabsorbed here. Okay, loop of Henle is also the part of nephron that enters the medulla, if you remember from the previous drawing. So let's say here, this will be medulla, and here will be cortex of the kidney. Okay, so now on to the next part, that will be the distal convoluted tubule. So distal tubule also comes really close to both the Bowman's capsule and efferent arteriole. I will tell you why. Okay, and then after coming close to the efferent arteriole, it will join with the collecting duct. But collecting duct is not part of the nephron, because many nephrons share one collecting duct. So now you have to look at it. Second reason why collecting duct is not considered part of nephron is that actually it has different embryologic origin. Okay. So this is distal tubule or distal convoluted tubule. Distal convoluted tubule. This is collecting duct. Collecting duct. Okay, so now we know all the parts of the tubule, of the nephron. So first, the Bowman's capsule, then the proximal convoluted tubule, then the loop of Henle, and then the distal convoluted tubule. These are the 4 parts of nephron. This is what you might be tested upon. Also, you might be tested upon that loop of Henle actually dips into the medulla. Okay, so now about the distal convoluted tubule. Again, same idea. You get water and ion reabsorption here. However, I would note that calcium also is reabsorbed here. So yes, you are losing the serum calcium here at the glomerular filtration membrane. You have to reabsorb it unless you want hypocalcemia. So that what is reabsorbed here, and this is pretty much it for the nephron. It's like general structure and basic function. Here in the collecting duct, I would just add one thing. Even though collecting duct is not part of the nephron, it still can reabsorb some stuff from the urine, from the forming urine, and it's water. And this is actually really important. I'm going to explain why in a little bit.